So good evening everyone. Today is the 9th problem solving session for the course Biochemistry. The course instructor is Professor Swagata Dasgupta from IIT Kharagpur and I am Shrija Ghosh from IIT Kanpur. So today's session is all about vitamins, their structure, different types of vitamins. So we'll solve different questions based on that topic, and feel free to ask me any doubt at any point. You can either unmute your mic or you can type in the chat box. So okay, so let's start with the questions. So the first question is, identify the correct statement or statements. First statement is, all water soluble vitamins except vitamin C are converted to cofactors. Second statement is, all water soluble vitamins except vitamin K are converted to cofactors. Third statement is, none of the lipid soluble vitamins except vitamin C are converted to cofactors. Fourth statement is, none of the lipid soluble vitamins except vitamin K are converted to cofactors. So we have to identify the correct statements here. So if anyone wants to answer, they can write in the chat box or they can unmute. So here the trick is to find out which vitamin, either vitamin C or vitamin K, is lipid soluble, and which one of those two are water soluble. If you know which is water soluble and which is lipid soluble, you will be able to answer. Okay, Akshara has answered option C. That is first and the fourth statement. That is, all water soluble vitamins except vitamin C are converted to cofactors, and none of the lipid soluble vitamins except vitamin K are converted to cofactors. So let's see what's the answer. Yes, option C is the correct answer. Vitamin C is one of the water soluble vitamin and vitamin K is one of the lipid soluble vitamin. So here is a list of water soluble and lipid soluble vitamins. So we have all the types of vitamin B, B1, B2, B3, B5, B6, B7 and B12, and vitamin C. These are water soluble vitamins, and rest of the vitamins, that is A, D, E and K, they are lipid soluble vitamins. Now among the water soluble vitamins, all the vitamin B types are converted to cofactors that attach to an enzyme or that work with the enzyme in order to catalyze its function. So these vitamins are basically not synthesized by our body, but we gain different types of vitamins from different food supplements or diet, from different kinds of diet. Okay, and once we get those vitamins are converted into cofactors, or those vitamins are directly involved in different enzymatic functions where an enzyme can catalyze its reaction with the help of these cofactors. So we'll see in the subsequent questions how each vitamin is converted into a particular cofactor and how those cofactors are actually helpful in catalyzing an enzymatic reaction. Okay, so among these water soluble and lipid soluble vitamins, you see vitamin C, that is a water soluble vitamin, is not converted into cofactor. It's actually a precursor to some other important biomolecules that we'll see in the further questions. Okay, and among the lipid soluble vitamins, so all these lipid soluble vitamins A, D and E, they are not converted to cofactor, but vitamin K is converted to cofactor. So in the question we had, all water soluble vitamins except vitamin C are converted to cofactor. So all the types of vitamin B are converted to cofactor, and none of the lipid soluble vitamins except vitamin K are converted to cofactor. So vitamin A, vitamin D and vitamin E, they are not converted to cofactor, only vitamin K is converted to cofactor. Okay, so just try to remember which of the followings are lipid soluble and which are water soluble, and among the water soluble ones and among the lipid soluble ones which are converted to cofactor and which are directly used for some kind of reaction or some kind of synthesis. Okay, so that is the main key point that you have to remember. So this is clear. We will move to the next question now. So question number 2. Which of the following statements about thiamine is or are incorrect? The active form of thiamine is a carboxylase. Thiamine is involved in the trans-ketolase reaction of triose phosphate pathway. Thiamine is involved in the dehydrogenation of beta keto acids, and magnesium deficiency has been shown to elevate thiamine deficiency. So which of the following statements are incorrect? Now you have to find the incorrect statement about thiamine. Okay. So in the previous slide I have shown you which vitamin is thiamine. What is it called? If anyone remembers. Thiamine is vitamin B1. Okay. And so now tell me which statements are incorrect here. If anyone knows the answer. So 3 of the statements are incorrect and there's only one correct statement. That's a hint for you if now someone can answer. Okay. Third statement is correct. Yes. Third statement is correct. No. The second statement is actually correct. So the answer is option C. So first, third and fourth statements are incorrect. Second statement, that is thiamine is involved in trans-ketolase reaction of triose phosphate pathway, that is the correct answer. So the active form of thiamine is actually a decarboxylase, and it does the active form of thiamine is actually called thiamine pyrophosphate, also known as TPP. Okay, TPP. This is the active form of thiamine, where 2 phosphates are attached to the thiamine structure. It is involved in the trans-ketolase reaction of the triose phosphate pathway. It is involved in the decarboxylation of the beta keto acids. We will see how it is doing the reaction. And fourth statement is also incorrect. Magnesium deficiency has been shown to alleviate the thiamine deficiency. Okay. So this is the structure of thiamine. So if you remember from the session about nucleic acids, if you remember the structure of purines and pyrimidines, can you see similarity of this ring with any one of those? Is it similar to purine or pyrimidine? Do you remember the structure of purine and pyrimidine? One had 2 ring system and one had a single ring system. So this one has a single ring system, which is similar to So if you see, the single ring system is actually similar to pyrimidine structure. So pyrimidine ring system. So thiamine has a pyrimidine ring system and then a side chain attached, and to this OH group. So this OH is reactive and we can attach different groups to this OH, right? So if I attach a pyrophosphate, that is two phosphates, sorry, if I attach two phosphate groups to this OH, what will we get? We will get thiamine pyrophosphate. Why pyrophosphate? This group is called pyrophosphate. Okay. So this group is called pyrophosphate. Okay. So, and from where thiamine is getting these two phosphates? It is getting from ATP. What was ATP? Do you remember what was ATP? ATP was adenine triphosphate. Adenine triphosphate. So, if you remember the structure of nucleic acids, nucleobases, we had adenine, thymine, cytosine, guanine and uracil, right? 5 bases we had, out of which uracil was found only in RNA and thymine was found only in DNA. Now, don't get confused between thiamine and thymine. Okay, in DNA we have something called thymine, and in and this thiamine, it is a vitamin. So this is a nucleobase, whereas this thiamine, it is a vitamin. Okay. Okay, so there is a difference between the two of the groups. This has a amine group attached to it, and that's why it was named thiamine, and it was actually one of the first vitamins that was discovered, and that's why the name vitamin came, amine, because of the amine group. It was earlier thought that all vitamins are amine containing molecules, but it was not true. Later it was found out that not all vitamins have amines, but the name came from vita, that comes from vital, that means life, because it's required for all kinds of living processes. So you cannot have life without vitamins, because vitamins are the precursors to many cofactors and many synthetic pathways that are required for life. Okay. So this adenine triphosphate, we have 5 nucleobases if you remember. So from that, adenine is attached to 3 phosphate groups. So adenine or adenosine triphosphate, actually it's not adenine triphosphate, it's adenosine triphosphate. So this is adenosine triphosphate. That means there is a ribose sugar, to which adenine is attached, and here we have 3 phosphate groups attached. Okay, and out of these 3 phosphate, these 2 phosphates are transferred to thiamine. Here you can see, if I remove these 2 phosphates, what will we left with? We will have this adenine or adenosine monophosphate, adenine combined with a ribose sugar and a phosphate. So adenosine monophosphate, this is triphosphate, this is monophosphate, and 2 phosphates are given to thiamine to become thiamine pyrophosphate. Okay, so this is the active form of thiamine. So this is the cofactor that is derived from thiamine. Okay, this is the cofactor that is derived from thiamine. And now what was the next statement? So active form of thiamine is thiamine pyrophosphate. It is involved in trans-ketolase reactions, that is true. It is involved in dehydrogenation of beta keto acids, that is false. It is actually involved in decarboxylation, not dehydrogenation, decarboxylation. Okay, how it is doing decarboxylation we will see. So you see here pyruvate. What is pyruvate? We have C double bond O, sorry, COH, then a C double bond O, and then CH3. So 3 carbons are there in pyruvate, and this pyruvate, from this pyruvate, if you remove a CO2, if you remove this group, the group highlighted here, if you remove this group, what we will be left with? We will be left with acetaldehyde, and this reaction is catalyzed by pyruvate decarboxylase, and this pyruvate decarboxylase actually requires TPP for the reaction to occur. Okay, pyruvate decarboxylase requires TPP as a cofactor for the decarboxylation of pyruvate to form acetaldehyde. Okay, to remove this CO2, to break this bond and to remove this CO2 from this complete molecule, this enzyme requires TPP as a cofactor, as a helper. Okay, TPP will provide the sufficient energy to break this bond and to release the CO2 from the pyruvate. Okay, and the resultant product will be acetaldehyde. When we will do bioenergetics, we will see why, where these reactions are important. Okay, for now you just remember that thiamine, which is vitamin B1, it is converted into TPP, which is the active form of thiamine, which is a cofactor which is important in decarboxylation and transketolase activity. Okay, it is important in 2 kinds of reaction, transketolase reaction and decarboxylation reactions. Okay, this is clear. If you have any form of doubt, you can ask, otherwise we will move ahead. So moving to the next question, question number 3. Identify the correct statements with regards to vitamin D. First statement is it has 2 major precursors forms, 7-dehydrocholesterol and ergocholesterol. Second statement is UV irradiation produces cholecalciferol and ergocalciferol. Third statement is vitamin D is neither a vitamin nor a cofactor, it is actually a steroid hormone. So we have to identify the correct statements here. So which are the correct statements about vitamin D? Which of the following are correct statements about vitamin D? Yeah, Akshara is saying all of them. That is true. All the statements are correct actually. So correct answer is option D, where all the statements are correct. So it has 2 major precursors. So vitamin D is actually converted from these 2 precursors. These 2 precursors are cholesterol. So if you remember the structure of cholesterol, we had a steroid nucleus and then an aliphatic chain was attached when we were studying fatty acids. In that section, if you remember, we had studied the structure of cholesterol. So this is 7-dehydrocholesterol. From the 7th carbon, hydroxy group is removed. So 7-dehydrocholesterol, when this is irradiated by UV light, we get, vitamin D is actually synthesized. So vitamin D is not directly found in nature, but from these different molecules, when these different molecules are irradiated by UV radiation, these molecules are converted into vitamin D, which is used by our body. That's why if you remember, it is always said that in the morning you should expose yourself to sunlight for vitamin D. Okay, people who are not exposed to sunlight for a long time, they tend to have vitamin D deficiency. Okay, so why do we get vitamin D deficiency? Because we cannot have vitamin D directly in our dietary supplements. All the other vitamins are found in some or the other food items, and then we eat those those particular food articles, and then we get those particular vitamins. But in case of vitamin D, we have to have these precursors of vitamin D, and we have to expose ourselves to UV radiation, not a high amount of UV radiation, but then some amount of sunlight in order to convert these precursor molecules into the vitamin D, which is required by our body. So vitamin D is actually not a vitamin or a cofactor, it is actually a steroid hormone, because it has a steroid nucleus. You remember the cholesterol structure, so it has a structure similar to cholesterol basically, because it's a because cholesterol type of cholesterol is a precursor of vitamin D. Okay, so 7-dehydrocholesterol, when irradiated by UV light, it gets converted into vitamin D, that is cholecalciferol, or even when ergocholesterol is irradiated by UV light, it gets converted into ergocalciferol, and then this is the active form of vitamin D. Just a second. So this is the active form of vitamin D. So this is how vitamin D is synthesized. So it's not a vitamin or a cofactor, it's actually a steroid hormone, and it has two precursors. So you have to remember the names of the precursor at least, and you have to remember that vitamin D cannot be gained by any dietary supplements. You have to get sunlight exposure in order for our body to synthesize vitamin D. Okay. I hope this is clear. So moving on to the next question, question number 4. Given structure is a derivative of a base. Options are A. adenine, B. guanine, C. cytosine, and D. uracil. Which of the following bases are there in this whole structure? If you remember the nucleobases, we had adenine, guanine, cytosine, uracil, and thymine. So which of the following base is there in this whole structure? Anybody knows the answer? Come on, we have already gone through nucleic acid. We have discussed the structures of nucleobases, what are nucleoside, nucleotide. So you should be able to answer this question. Nobody? The correct answer is adenine. So the structure is derived from adenine. This base here you see, so this is the ribose sugar, this is the phosphate, and this here, this structure, this is a nucleobase. This structure is called a nucleobase. This is the sugar and this is the phosphate. Okay, and this nucleobase is adenine. So this is the structure here. We have adenine, guanine, uracil, thymine, and cytosine. So see, these two are purines and these three are pyrimidines. Purines have two ring system. A 6-membered ring is fused with a 5-membered ring, and pyrimidines have a single ring system. And if you see here, in adenine we have this NH2 group attached at the 6th carbon, whereas in guanine at that same carbon we have a carboxyl oxygen along with a additional amino group at the 2nd carbon. So this is guanine. So this is very distinctive from each other. So you can differentiate between adenine and guanine easily by seeing the amino group at the 6th carbon here. So most of the time you will see, if you see a 2-membered 2-ring system, double ring system as a nucleobase, you know it's either adenine or guanine. Now all you have to do is you have to check whether here we have a amino group or a double bond O. If we have a double bond O and an amino group here, that is guanine. If we have only amino group at this position, this is adenine. If we have single ring system, then we have to identify between uracil, thymine and cytosine. So at least you should be able to reach to the answer by knowing whether it's a purine or pyrimidine. So if you know the difference between purine and pyrimidine, half of your problem is solved. So if you already knew that this is a purine ring system, so that means 2 options are already eliminated. Now among these 2 options, now you have to see the side chain, what is there, what is the substitution there, and that's how you will find the correct answer to be adenine. Okay. So try to understand the structure. I am not asking you to learn all the structures, it's not possible, but then try to figure out, at least you should be able to see a structure and then think like, yes, this could be adenine. If I ask you to draw adenine and you don't draw it correctly, that's fine. But if I show you the structure of adenine, at least you should be able to identify that. Okay. So you have to try to understand what is try to find one particular feature about each structure, so that you see a structure and you can say, okay, this is adenine. In case of vitamins also, you have to at least see what is the base of the structure. Like for thiamine, we had a pyrimidine ring system only and a side chain was attached, and then when a pyrophosphate is attached to that thiamine structure, we get the active form of thiamine. So like that, all the vitamins have a distinctive structure which you should at least able to identify, okay, this is this vitamin and what is the role of it. For thiamine, we know the role is to do decarboxylation and ketolase reactions. Okay, so like that we will try to remember the function of each vitamins. Okay. So moving to the next question, question number 5. The labels A, B, C are respectively. So here in the structure, what is A, what is B and what is C? Option A nucleotide, B beta and glycosidic bond and C nucleoside. Option B, A nucleoside, B beta and glycosidic bond and C nucleotide. Option C, A nucleotide, B is alpha and glycosidic bond and C is nucleoside. Option D is A is nucleoside, B is alpha and glycosidic bond and C is nucleotide. So what would be the correct answer? This also we have studied in the previous session when we were discussing nucleic acids. So you should be able to answer this, which is called nucleotide and which is called nucleoside. Okay, Akshara has answered option A. that is, so the complete structure is nucleotide. This bond is beta and glycosidic bond, and the structure without the phosphate, ribose and the base is nucleoside. Okay, that is the correct answer, option A. So when you have, so you have basically a phosphate group, a sugar and a base. This is called nucleotide. If I remove the phosphate from it, if I only have the sugar and a base, it is called nucleoside. And the bond between sugar and the base, so any bond that is formed with the sugar, it is called a glycosidic bond. So when we will study carbohydrates in the next session, we will see what is a glycosidic bond. So here the bond is with a nitrogen, that's why it's called an N-glycosidic bond. And about the alpha and beta glycosidic bond also we will see in the next session, how a sugar can form alpha glycosidic bond and how a sugar can form beta glycosidic bond. Okay, so these are the things that we will learn in the next session when we will discuss the structures for carbohydrates. For now, just remember that any bond between sugar and another atom, that bond is called a glycosidic bond, and since the bond is with a nitrogen, it is called an N-glycosidic bond. If the bond was with this oxygen, it would have been O-glycosidic bond. Okay, if the bond was between carbon and carbon, it would have been a C-glycosidic bond. Okay. Okay, so the next question here, the given structure belongs to a certain family of molecules. Which of the bases stated below is not expected to be the part of the said family of molecules? This structure is part of a family of molecule. Which of the following bases or which of the following molecules are not expected to be part of that family of molecules? So what would be the answer? Do you understand the question? Uracil and thiamine. Yes, Akshara has answered here, uracil and thiamine. So do you know why this is the correct answer? So if you see here in this sugar, you have to observe this particular group. So we have only hydrogen here. So you remember we had two types of molecules. We had a ribose sugar and a deoxyribose sugar. Deoxyribose sugar. So ribose was part of RNA and deoxyribose was part of DNA. So if there was a OH here, it would have been a ribose sugar. Since this O is not there, this is a deoxyribose sugar. And now this deoxyribose sugar is part of the family DNA. And now we know uracil is not found in DNA, it is only found in RNA. And thiamine is actually not found in any of the DNA or RNA molecules. It is not a part of nucleic acid, it is a part of vitamin. Okay, it is a type of vitamin which is converted to a cofactor which is used in different enzymatic reactions. So this is not part of nucleic acid at all. So thiamine is not part of this nucleic acid family and uracil is not part of the DNA family. So uracil is part of nucleic acid, but it is part of the ribose nucleic acid. It is not part of the deoxyribose nucleic acid. Okay. So correct answer is third and the sixth bases, that is uracil and thiamine. Okay. So I hope this is clear. So you have to see the difference also between these two. Thymine and thiamine, these are two different. This is a vitamin, this is a nucleobase. Okay, moving to the next question. Identifying A and B, state the vitamin that is involved in the reaction. So we have to tell what is A, what is B, and which vitamin is involved in the reaction. So does anyone know the answer? Which of the following is A, which of the following is B? So option A is A FAD, B FADH2, and the vitamin that is involved is riboflavin. Option B we have A is FAD, B is FADH2, and the vitamin involved is cobalamin. Option C is A is FADH2, B is FAD, and vitamin involved is riboflavin. And option D is A FADH2, B FAD, and vitamin involved is calciferol. So which of the following is the correct answer? Option A, yes. So option A, that is A is FAD. So FAD is reduced to FADH2, and the vitamin involved is riboflavin. So riboflavin is basically precursor for FAD and FADH2. So the active form of riboflavin is FAD. So like you have seen in case of thiamine, the active form was TPP, thiamine pyrophosphate. Similarly, for riboflavin we have the active form as FAD. Okay. So riboflavin is vitamin B2. Okay. So correct answer is option A. So here you see, this is the structure of FAD. So this part here, this part is riboflavin. Okay, this part here is riboflavin. Then this riboflavin, what is FAD? FAD is flavin adenosine dinucleotide. So flavin adenine or adenosine dinucleotide. Why it is called dinucleotide? Here you see. So this is a adenine molecule. This is the ribose sugar. Okay, and this is the phosphate. So this here we have AMP, right? And this AMP is attached to another structure via phosphodiester bond. So it makes a dinucleotide. You remember, two nucleotides attached by a phosphodiester bond, they are called dinucleotides. So here this is the second nucleotide. Here we have riboflavin that is attached to a phosphate group, which is again attached to another phosphate by a phosphodiester bond. This phosphate is attached to a ribose sugar and adenine. So we have flavin, that is riboflavin, adenine, dinucleotide. Okay, and here the two nitrogens in the riboflavin ring system, these two nitrogens have extra lone pair. So when this oxidation reaction is happening, so here what is happening? Dehydrogenation is happening, right? So we have here 2H, two hydrogens are there and two hydrogens are here, right? Both of these carbons have two hydrogens. From these two hydrogens, one hydrogen is removed from each carbon. So two hydrogens are removed. So we are left with one hydrogen here and one hydrogen here. So two hydrogens are removed. So who will take the two hydrogens? This FAD molecule will take. So these two nitrogens, they have they can attach two hydrogens, so they become FADH2. So basically a reduction reaction is happening here. FAD is the oxidized form, it is reduced to FADH2. Okay, this is FADH2. Two hydrogens are attached to FAD to become FADH2. This is the reduced form. Okay, so here you can see two electrons are added, so that means reduction is happening. The reverse reaction would be an oxidation where electrons are removed. Addition of electron is reduction, removal of electron is oxidation. Okay, so here succinate molecule, it is dehydrogenated, two hydrogens are removed here, and those two hydrogens are given to FAD to become FADH2, and FAD is basically the active form of riboflavin. Riboflavin is the vitamin B, vitamin B2. Okay, so riboflavin, from riboflavin we got FAD. FAD will take up two hydrogens to become FADH2. Okay, and you understood why it is named FAD, why flavin adenine dinucleotide, because it has a flavin molecule and it is added to the adenine nucleotide by a phosphodiester bond, so it becomes two nucleotides, so dinucleotide. Okay, this is clear. So the next question, question number 8. Identify the correct statement regarding riboflavin. Again, so options are: A, it is the vitamin B6, B, it occurs as the white pigment in eggs and milk, C, it turns colorless on reduction with zinc in acid and regain its color upon reoxidation, D, it turns deeper in color on oxidation, regain its color on reduction with zinc and acid. So which of the following statement is correct about riboflavin? Come on, tell me which of the following statement is correct about riboflavin? It is vitamin B6, it occurs as the white pigment in eggs and milk, turns colorless on reduction with zinc and acid and regain its color upon reoxidation, or it turns deeper in color on oxidation and regain its color on reduction with zinc and acid. What would be the correct answer? Anyone knows? So the correct answer is option C, it turns colorless on reduction and regain its color upon reoxidation. Why does it happen? So if you see the structure of riboflavin or FAD, so riboflavin, I told you it's not vitamin B6, it is vitamin B2. So first statement is incorrect. It does not occur in eggs and milk, the white pigment on eggs and milk, it is not riboflavin. Third statement, that is the correct statement, it turns colorless on reduction and regain its color upon reoxidation. Why it happens? So if you see this ring system of riboflavin, it imparts a yellowish color due to these nitrogen. Due to the presence of these nitrogens, it imparts a yellow color. Okay. Due to presence of these nitrogens, it imparts a yellow color to the molecule. When it is reduced, it becomes FADH2, where these two nitrogen, the lone pairs of these two nitrogen are now involved in bonding with hydrogen. Okay. So when the lone pairs were available, they were imparting a yellowish color to the molecule. Once it is bound to hydrogen, the molecule cannot impart any color, thus becomes colorless. So this molecule is colorless, whereas this has a yellow color. Okay. this has a yellow color because of these two nitrogens. So when FAD is actually, or riboflavin in that matter, is reduced with zinc, when zinc is used for the reduction of riboflavin, it becomes colorless, because the reduced form of riboflavin is colorless, and when reoxidation is done, it again regain its color because the hydrogens are removed. When again the reverse reaction will be done, these hydrogens will be removed and it will turn into riboflavin that has a yellow color. Okay, so it will regain the color. Okay, so when the reduction with zinc will happen, zinc will turn into Zn2 plus, and then again it will come back to Zn0 state and riboflavin can regain its color. Okay, the correct answer is option C. This is clear how riboflavin has the color. Okay. Moving on to the next question, question number 9. The structure shows adenosine triphosphate. It is an important and versatile molecule that can donate the X, Y and Z moieties. Name the Z moiety. So now here you see we have X, Y and Z. So we have to name what is Z here. So we have just now discussed what base is this. First tell me. We have a base attached to a ribose sugar and then phosphates attached. Okay, Akshara has answered option D, AMP. Okay, yes, Z is AMP, that is true. So this base is adenine. Here we have adenine attached to a ribose sugar and 3 phosphates are attached. So the complete molecule is actually ATP. This complete molecule is ATP. From this ATP, if we remove these 2 phosphates that are marked with red box, this Y, what is this Y? This Y is basically pyrophosphate. 2 phosphates together are called pyrophosphates. So Y is the pyrophosphate and X is monophosphate. So when Y is removed from ATP, it becomes AMP. When X is removed from ATP, it becomes ADP. So if I ask you what is this part, this will be ADP. Okay. So adenosine monophosphate, adenosine diphosphate and adenosine triphosphate. Okay. So this is a nucleotide. Okay, this is a nucleotide because we have an adenine, we have a ribose sugar and we have phosphate. So it's a nucleotide. Okay, so ATP, ADP and AMP. This part is called pyrophosphate, Y is pyrophosphate and X is monophosphate. Okay, or X is also called gamma phosphate. Why? Because this is attached to the carbon directly. So this is alpha, then this is beta and this is gamma. So X is called gamma phosphate, this phosphate is called beta phosphate, this phosphate is called alpha phosphate. Okay, so if gamma phosphate is removed from ATP, it will become ADP. Then the next phosphate, beta phosphate, will be removed, it will become AMP. This is clear? So correct answer is option D. Okay, moving to the next question. Question number 10. While eating cooked eggs is highly nutritious, eating raw eggs is advised against. Given that the raw eggs contain avidin, what may be the possible reason for this? Option A, avidin binds tightly to riboflavin, making riboflavin less available for its enzymatic functions. Option B, avidin cannot be digested by the gut bacteria. Option C, avidin binds tightly to folate, making folate less available for its enzymatic functions. And option D is avidin binds tightly to biotin, making biotin less available for its enzymatic function. So, which of the following options are option is correct? What is the reason behind not eating raw eggs? So, it is always advised to cook your eggs properly before eating them. Okay, we have the answer option D. Avidin binds tightly to biotin, making biotin less available for its enzymatic action, which is the correct answer. So, biotin is again another vitamin, and this vitamin is involved in certain enzymatic functions. We will see how biotin is involved in the enzymatic function. But what happens is avidin has such a high affinity to biotin that biotin is not available for the enzyme to bind. Avidin will attract biotin towards itself and the person can have biotin deficiency. Okay. So, biotin supplements need to be taken in case of high amount of avidin present in the body. So, that is why it is advised not to eat raw eggs. Generally, when if you are consuming boiled eggs, it is always advised to boil the egg properly. Hard boiled eggs are advised by the doctors to be eaten, because raw eggs contain these avidin and it can actually cause biotin deficiency in the body, because whatever biotin will be there in your food will be taken up by the avidin. The biotin will be tightly bound to the avidin and there won't be any free biotin available for the enzyme that requires biotin. For those enzymes, there won't be any free biotin available in the body, and thus those enzymes won't be able to perform its function without the biotin. Okay. So, avidin can cause biotin deficiency in the body, and that's why it is advised not to eat raw eggs. Okay. This is clear? Till now, if anyone has any doubt, please ask. So, we will move to the next question. Question number 11. Given is the structure of a metabolite of vitamin A1. Given is the structure of a metabolite of vitamin A1, it is essential for cellular differentiation, controlling cellular growth. Identify the compound. So, you have to identify this compound. This is a metabolite of vitamin A. So, what is this compound? What would be the correct answer for this? Option A, riboflavin, option B, niacin, option C, retinoic acid, and option D, vitamin B6. So, what is the structure shown here? Structure shown here is a metabolite of vitamin A. So, if you know what is vitamin A, you will be able to answer this question very easily. Come on, tell me what would be the answer? Till now we have discussed about riboflavin, niacin and vitamin B6. So, automatically you know that none of these are vitamin A. So the correct answer, logically you can answer that the correct answer is retinoic acid, right? Correct answer is option C, retinoic acid. So this is a metabolite of vitamin A, that is retinol. It is derived from vitamin A. So here we have the carboxylic acid group. So that's why it is called retinoic acid. Retinol will have a alcohol group, that is OH. Okay. So this is clear. So this is about vitamin A1. So till now we have studied about thiamine, that is vitamin B1. Then we have studied about riboflavin, vitamin B2. We have seen vitamin A. We have seen vitamin D, the role of vitamin D. Okay, we have studied about biotin, that is vitamin B7. So try to remember about each vitamin, so that's how you will be able to learn the basic structure of vitamin and which vitamin is required for what function and like that. Okay. Again we will study about the next vitamin here. Question number 12. Given that dopamine is a amine neurotransmitter, name the vitamin that helps in the process shown. So here we have a process where phenylalanine, which is an amino acid you have studied, it is converted into tyrosine. Then tyrosine is converted into dopa, that is dihydroxyphenylalanine. So you see this phenylalanine has 2 OH and this is called dihydroxyphenylalanine, and this dopa is now converted to dopamine, where this CO2 is removed from dopa and we have a structure called dopamine, and dopamine is actually an amine neurotransmitter. Okay. So there is a vitamin that is involved in this whole process. So we have to name the vitamin. So we have the option vitamin B5, B6, B7, B9. What would be the correct answer here? Yeah, we have the answer. Akshara has answered vitamin B6. Okay. So vitamin B6, what is the name of vitamin B6? If you can write. Do you know the name of vitamin B6? You are correct. The correct answer is vitamin B6. So vitamin B6 is pyridoxine. Okay. Pyridoxine is vitamin B6. It is actually converted to pyridoxal and pyridoxal phosphate. So here in pyridoxine we have an alcohol group. Okay. This alcohol group is converted to aldehyde group and the molecule is called pyridoxal or pyridoxaldehyde. Okay, in short we call pyridoxal. This pyridoxal, then to this pyridoxal a phosphate group is attached at this position, and this phosphate is achieved from a ATP molecule. So ATP will become ADP and a phosphate will attach to this pyridoxal. So it will become pyridoxal phosphate, and this pyridoxal phosphate is actually involved in the reaction of decarboxylation. So here if you see, this R group is nothing but this part. Okay. So a carbon is attached to a carboxyl COO and an amino group. So this is a basic amino acid structure, a carbon attached to a hydrogen, C double bond O, NH3 and a side chain. So this is the side chain, R group. Okay. So this part is R group. Okay. So here if you see, we have carbon, amino group, COO and R group. So from this when pyridoxal phosphate reacts with this, what it does, it basically makes a complex where it takes up the amino group towards amino group along with it and becomes pyridoxamine phosphate. Okay, pyridoxamine phosphate, and in the further reaction it can actually help in decarboxylation of this product. So dopa can be converted into dopamine, and this decarboxylation aromatic amino acid decarboxylase, this enzyme requires vitamin B6, that is pyridoxine, or modified version of pyridoxine, that is pyridoxal. Okay, pyridoxal phosphate, that is the active form of vitamin B6. So you remember the active form of vitamin B1 was thiamine pyrophosphate. Okay, active form of vitamin B2 was flavin-adenine dinucleotide. Similarly, for B6 we have pyridoxal phosphate. Okay, pyridoxal phosphate we have. Okay, so B6 is pyridoxine, and from pyridoxine we get pyridoxal phosphate, and this pyridoxal phosphate is actually helps in the decarboxylation. It takes out the CO2 and it helps in the decarboxylation reaction and amino acid synthesis. Okay, mainly it is involved in amino acid metabolism. When we will study amino acid metabolism, you will see the role of pyridoxal phosphate there. Okay, so TPP was also doing decarboxylation, but it was doing decarboxylation of the beta keto acids. Pyridoxal phosphate is doing the decarboxylation of amino acids. So that is the difference between the two. Okay, all right. So I hope this is clear. Okay, next question, match the following. So we have different names of vitamin and their so the chemical name of vitamins and the vitamin names here. So we have ascorbic acid, biotin, thiamine and cobalamin, and we have here vitamin B1, B7, B12 and C. So we have to match. So what would be the correct answer here from all the 4 options? Try to match and then let me know the correct answer. Come on, what would be the correct answer? Okay, Akshara has answered option D. Okay, let's see if option D is correct answer here. So ascorbic acid, what is ascorbic acid? It is vitamin C. Okay, so A4. Okay, correct. Then biotin, biotin is vitamin B7. Okay, so B2, that is also correct. Thiamine, thiamine is vitamin B1. Okay, so C1, and cobalamin is vitamin B12, so D3. Yes, so option D is the correct answer. Yes. Okay, so we have vitamin C as ascorbic acid, vitamin B7 as biotin, vitamin B1 as thiamine and vitamin B12 as cobalamin. Okay, so just for the revision we will go through this once again. So we had water soluble vitamins and lipid soluble vitamins. From these Vitamin B1 is thiamine, B2 is riboflavin, B3 is niacin, B5 is pantothenic acid, B6 is pyridoxal or pyridoxine, B7 is biotin, and B12 is cobalamin, and vitamin C is ascorbic acid. What is vitamin D? Vitamin A, we have studied. Vitamin A is retinol. What is vitamin D? Vitamin D, we have precursors of vitamin D that are forms of cholesterol. Okay. So, that was ergocholesterol and 7-dehydrocholesterol, and we will study about vitamin E and K also. So, among these we remember thiamine, that is vitamin B1, the active form is TPP, the cofactor is TPP, involved in decarboxylation of beta keto acid and transketolase reaction. Vitamin B2 riboflavin, the cofactor is FAD, involved in the redox reaction where FAD is converted into FADH2. FAD is reduced to FADH2. Okay. We haven't studied about niacin yet. We will come to the question about niacin pantothenic acid. Then we have studied about pyridoxine or pyridoxal. This gets converted into pyridoxal phosphate and it is involved in amino acid biosynthesis pathway, amino acid metabolism. Okay, amino acid metabolism. Then we have biotin. We remember biotin has a affinity towards streptavidin, and biotin is actually involved in different enzymatic reaction. We will see biotin, a role of biotin also. Cobalamin is also an important vitamin. We haven't gone in detail for cobalamin here. And vitamin C, you all know, ascorbic acid and folic acid. We will see what role of folic acid is. Retinol also we have seen, and how vitamin D synthesis is happening by UV radiation of different precursors cholesterol, that we have seen. Okay. So, till now we have gone through these molecules. We will go through niacin, pantothenic acid and biotin and folic acid now. Okay. Till now, any doubt, in any of the vitamins that we have discussed, any doubt in those vitamins? If you have any doubt, you can ask now, so that before proceeding I can clarify. Okay. So, the next question, sorry. Identify the DNA bases derived from the structure shown. So, here we have a structure of folic acid, and from this folic acid actually two bases of DNA are derived. Which are the two bases? So, if you see here, you will see a ring system. So, you can identify what bases are actually derived from folic acid. Option A, thiamine and uracil. Option B, adenine and guanine. Option C, adenine and uracil. Option D, thymine and guanine. Okay, we have the answer option B, that is adenine and guanine. Yes. So, adenine and guanine are purines, right? And folic acid is actually involved in purine synthesis. Okay. So, if we have folic acid deficiency, it can actually cause cell death, because there won't be any DNA synthesis and that will lead to cell death. So, in case of cancer, when they have to kill the cells, because cancer is basically undifferentiated cell growth, right, uncontrolled cell growth. So, at that time we have to stop the cell growth. In those cases, folic acid is actually inhibited. Okay, we give drugs that can inhibit the purine synthesis pathway. So it is involved in purine synthesis pathway. So when this pathway is inhibited, cell growth is automatically stopped. Okay, so this is a very important molecule in our body. Without this, life won't be possible. Without this molecule, without this folic acid, life won't be possible because it is involved in the basic synthetic pathway in the body. Without DNA, there won't be any life. Your DNA contains the genetic information, all the processes that has to happen in your body. So if a cell does not have proper DNA, the cell cannot survive. Okay, and folic acid is involved in the purine synthesis. That means it is involved in synthesis of adenine and guanine. So without A and G, you cannot have a proper DNA and you cannot have a proper cell. Thus life won't be possible without folic acid. Okay, this is clear. And folic acid is actually vitamin C, part of vitamin C. Okay. So question number 15. Considering the reaction scheme provided above, state the product form. So we have here thiamine and we have TPP synthetase, ATP and AMP. We have to tell what is the product form. So we have already discussed this. So tell me what would be the product form. Option A, thiamine phosphate. Option B, thiamine adenosyl phosphate. Option C, thionin adenylyl pyrophosphate, and option D is thiamine pyrophosphate. What would be the correct answer here? Option D, that is thiamine pyrophosphate. Yes, Akshara has given the correct answer. Option D, that is thiamine pyrophosphate. So thiamine will take two phosphates from ATP. ATP will get converted into AMP because two phosphates are removed. So triphosphate will become monophosphate, and those two phosphate will attach at this position to become thiamine pyrophosphate. Okay. So you see here we have thiamine, we have TPP synthetase. So you don't have to mug up the structure or mug up the names of the enzyme. So you see what is happening here. TPP is being synthesized. So what would be the enzyme? TPP synthetase, because this enzyme is synthesizing TPP. Okay, this is TPP. So we have the OH here. At this oxygen we have two phosphate groups attached. A pyrophosphate group is attached to become TPP. Okay, till this part we have thiamine. Till this part we have thiamine, and then thiamine has two pyrophosphates attached, that is, and these two pyrophosphates are coming from ATP molecule. ATP is losing that pyrophosphate to become AMP molecule. Okay, simple. Okay, next question here. Which of the following is a component of coenzyme A? So here we have to answer which of the following is component of coenzyme A. Option A, retinol. Option B, pyridoxine. Option C, retinoic acid, and option D, pantothenic acid. Which of the following is a component of coenzyme A? Tell me what would be the answer. What is the component of coenzyme A? Retinol, pyridoxine, retinoic acid, and pantothenic acid. Okay, Akshara has answered option D, pantothenic acid. That is the correct answer here. So we have studied retinol. What is retinol? Retinol is vitamin A. Pyridoxine, what is pyridoxine? Which vitamin is pyridoxine? It is vitamin B. What is pyridoxine? Come on, that is how you will remember. Pyridoxine is vitamin B6. Okay. Retinoic acid is derived from retinol only. So it is part of vitamin A. Pantothenic acid, what is pantothenic acid? It is vitamin B5. Okay. So pantothenic acid, here we will see. So we have this coenzyme A, which is again a cofactor. So pantothenic acid is a part of this coenzyme A. So here you see coenzyme A has the adenine base attached to a ribose sugar. This ribose sugar is again attached to a phosphate group at the 3' position, and the pyrophosphate group is attached. So this is basically ADP along with a phosphate at the 3' of ribose. So this is for 1', 2', 3', 4' and 5', right? 5 carbon sugar. This is ribose, 5 carbon sugar, and at the 3rd carbon we have a phosphate group attached, and we have 2 phosphate groups attached to the whole molecule. So this is ADP along with a phosphate, right? To this ADP, now from this phosphate you see pantothenic acid is attached, and from this pantothenic acid we have here cytosine attached, cysteine attached. So you remember the amino acid cysteine that has a SH group. So this is cysteine amino acid that is attached to pantothenic acid and ADP along with a phosphate at the 3' position. So this is the structure of coenzyme A. Where this coenzyme A is required? Anybody knows? Why do we need coenzyme A? So coenzyme A is basically mainly used for transfer of acetyl groups. So any form of alkyl groups can be transferred along with coenzyme A. So the most common molecule that is present in the biosynthetic pathways, bioenergetic pathway in our body, it is acetyl coenzyme A. So whenever 2 carbons have to be transferred together. So for transfer of 1 carbon, we have many molecules, but for transfer of 2 carbons together. So what is acetyl? Acetyl is basically CH3CO. So for transfer of the CH3CO to any molecule, acetyl CoA is the molecule that does the transfer of acetyl groups. Okay, so any acyl molecules are transferred by this cofactor, by the help of this cofactor. And from where we are getting this cofactor? We are getting this cofactor from the vitamin B5, that is pantothenic acid. Okay, so pantothenic acid is a part of this coenzyme A. So without pantothenic acid, coenzyme A will not be formed. Without coenzyme A, we won't have acetyl coenzyme A or malonyl coenzyme A for the transfer of those particular groups to a particular molecule. Okay, so when we will study citric acid cycle, you will see why acetyl coenzyme A is important. Okay. I hope this is clear. Moving to the next question, which of the following is involved in the carboxyl group transfer reaction? Which of the following is involved in the carboxyl group transfer reaction? So transfer of a carboxyl group: thiamine, biotin, pyridoxine and pantothenic acid. So what is involved in the carboxyl group transfer reaction? What would be the correct answer here? Option A thiamine, option B biotin, option C pyridoxine, and option D pantothenic acid. Right now we saw the role of pantothenic acid, so it's not transferring carboxyl group. What it is transferring? It was transferring acetyl groups or any acyl groups basically. We have seen the role of thiamine. What was thiamine doing? Thiamine was doing basically decarboxylation, right? We have seen the role of pyridoxine. So which of the following will do the carboxyl group transfer reaction? The answer is biotin. So for thiamine we have seen the active form is TPP and the role is decarboxylation reaction and transketolase reaction. Okay, you have to remember this. Thiamine, that is vitamin B1, active form is TPP, role is decarboxylation of beta keto acids and transketolase reaction. We have biotin here, that is vitamin B7. Biotin does carboxyl group transfer reaction. So it transfers the carboxyl group from one molecule to another. Okay. Okay. What is pyridoxine? Pyridoxine is vitamin B6. Okay. Pyridoxine is vitamin B6, and what is pyridoxine doing? It is doing the decarboxylation reactions in amino acid metabolism. It is basically involved in amino acid metabolism. Okay. And pantothenic acid, that is vitamin B5. What was pantothenic acid doing? It was part of coenzyme A, the cofactor coenzyme A. So for each vitamin we know the active form of vitamin and we know the role of vitamin. That is important. You have to remember the name of the vitamin, the role of the vitamin, and the active form of the vitamin that is performing that role. Okay, none of the vitamins are directly involved in the reaction. They are always converted into something else and then doing the reaction. So all the vitamin B, especially the vitamin B, they are converted into cofactor before performing a particular reaction. Only vitamin C is not converted into any cofactor, and among the lipid solubles vitamin A, D and E are not converted into cofactors. Okay, rest all the vitamins are converted into cofactors or active forms before they are involved in any kind of reactions. Okay. So how is biotin doing this carboxyl transfer? We will see. So this is the structure of biotin. So remember we had studied earlier, biotin has an affinity towards avidin. Avidin, that is found in raw egg. Raw eggs have avidin, and avidin can bind to biotin. Avidin binds to biotin and causes biotin deficiency, right? This is the information that we had studied earlier for biotin. Now we have another information about biotin, that is vitamin B7. Another information about biotin is it does carboxyl group transfer reaction. So, it can transfer the carboxyl group from one molecule to another molecule. How it will do that? For transferring a carboxyl group, first it requires a carboxyl group. So, biotin, the active form of biotin is actually carboxybiotinyl enzyme. Okay, so biotin is always attached to an enzyme by a peptide bond. So, you see here a carboxyl group is there. If we have COOH, if it has to make a peptide bond, how will it make? So, if I have RCOH and if I have if this is R1COH and this is R2NH2, if I want to make a peptide bond between these two, what will be how will we make the peptide bond? It will be basically C double bond O, NH and R2, right? So, similarly from this COOH, it attaches to the lysine residue of an enzyme. So, you remember the side chain of lysine? Do you remember the side chain of lysine? Lysine had 4CH2 and then an amino group. Okay. So, this was the structure of lysine, right? This was the structure of lysine. So, this amino group will be involved in the peptide bond formation with this carboxyl group, not the amino group of the amino acid. Why? Because in the enzyme, this amino acid is part of the enzyme. So, that means this amino and the carboxyl group are involved in the peptide bonding in the protein, right? So, protein is made of different amino acids. Those amino acids are bonded with peptide bond. So, these two groups will be involved in that peptide bond formation. The group that will be free for bonding will be the side chain amino group. The side chain amino group will form the bond with the COH of biotin. So, this is the bond that I am talking about. So, this is that free amino group. So, we have a CONH bond, that is a peptide bond, and now you have 1, 2, 3, 4. So, CH2 4 times, and then you have this ring system here. In this ring system, what biotin will do? Biotin will acquire a carboxyl group from a bicarbonate. Okay, from a bicarbonate, biotin will acquire a carboxyl group. So, you see a carboxyl group is here. This carboxyl group has been acquired by biotin from a bicarbonate. So, from bicarbonate, biotin will acquire a carboxyl group, and from this COOH, it will bind to the enzyme from a lysine residue. So, it will bind to the lysine residue of an enzyme, and carboxyl group will attach to this part of biotin, and the carboxyl group will come from a bicarbonate. Okay. And then once the carboxyl group is here, now biotin, the active form of biotin, carboxybiotin, it is now ready to transfer this carboxyl group to another molecule. Now, it has acquired a carboxyl group. So, it has taken a carboxyl group from bicarbonate and it will give it to pyruvate enolate. So, it is the enol form of pyruvate. Okay, this is the enol form of pyruvate, and it will transfer this carboxyl group to this pyruvate enolate. So, to this CH2 group, it will transfer the carboxyl group. So, you see this particular carboxyl group is acquired from biotin. So, I will just color code it for your ease. So, the carboxyl group highlighted here with green. This is this particular carboxyl group. Okay, this particular carboxyl group. And this is the already existing carboxyl group of pyruvate. Okay. So the resulting molecule that we are getting is oxaloacetate. Okay, so we have two carboxyl groups at the terminal ends and COCH2. Okay, so from pyruvate enolate we are getting oxaloacetate by a biotinyl enzyme. Biotinyl enzyme means the enzyme lysine residue of that enzyme is attached to the biotin. This biotin will acquire a carboxyl group from a bicarbonate and transfer. So this green carboxyl, this is acquired from bicarbonate and it is transferred to the pyruvate enolate to form oxaloacetate. Okay, so this is clear how biotin is involved in a carboxyl group transfer reaction. Okay, this is clear. Okay, moving to the next question. NAD and NADP are cofactors of which vitamin? So options are vitamin B1, B2, B3 and B5. So NAD and NADP, these two are cofactors of which vitamin? B1, B2, B3, B5. What would be the correct answer here? So what is B1? B1 is thiamine. I am telling you the names again and again so that you remember. Okay, you also when you are studying, whenever you see vitamin B1, try to remember, okay, which vitamin is that, or if you see the name thiamine, try to remember, okay, which what is the name of that vitamin. Okay, B2 is riboflavin, B3 is niacin, B5 is pantothenic acid, pantothenate. Okay, now tell me, NAD and NADP are cofactors of which vitamin? B3, yes. So option C is the correct answer. Niacin is the precursor of NAD and NADP. So this is very similar to riboflavin. So like riboflavin was converted into flavin adenyl dinucleotide. Similarly, niacin, also known as nicotine, this is converted into nicotinamide adenosine or adenyl dinucleotide. Okay, so this is very similar to riboflavin. The reaction that it catalyzes is also redox reaction. So we have niacin, that is nicotinic acid, sorry I was wrong, nicotinic acid. So the nicotinic acid, because we have the COH group, now this OH is replaced by an amine group to become nicotinamide. So this is a amide bond, right? CONH is a amide bond, so nicotinamide. Okay, now this nicotinamide, when it is attached to a sugar and a phosphate, it becomes a nucleotide. This is one nucleotide, right? This is one nucleotide, nicotinamide, ribose and phosphate. This is another nucleotide. This is what this is? This is adenine. This is adenine, ribose and phosphate. So this becomes another nucleotide. So that means the complete molecule is nicotinamide adenyl dinucleotide. Why dinucleotide? Because two nucleotides are attached by a phosphodiester bond. So dinucleotide. So nicotinamide adenyl dinucleotide is the molecule. Do you understand how this is dinucleotide? So instead of this OH, if we attach another phosphate here, what it will become? It will become NADP plus. Okay. So if I attach a phosphate, it will become NADP plus. Right now without phosphate it is NAD plus. From where we are getting the plus? Sorry. Yeah. So from where we are getting this plus? Yeah. So we are getting this plus. If you see here on this nitrogen, we have a positive charge. Okay. So we have a positive charge on this nitrogen. So this molecule is NAD plus. Okay. If we have a phosphate group at the 2' 2' carbon attached to this oxygen, if we eliminate this hydrogen and attach a phosphate, we will get NADP plus. So both NADP plus and NAD plus are involved in the redox reaction, very similar to FAD. So for FAD, what was the reduced form? FADH2. Similarly for NAD plus, the reduced form is NADH. Okay. So this is the oxidized form and this is the reduced form. Okay. So NAD plus is the oxidized form and NADH is the reduced form. So here we have one extra hydrogen attached, that plus charge is neutralized by conjugation, and we have one H plus released. So NADH plus H plus. So the reaction goes like NAD plus is reduced to NADH plus H plus. So what is happening? Loss of electron is happening. Loss of electron is happening here, right? Gain of proton is happening in this direction. So this is the reduction reaction. This will be the oxidation reaction. Okay. So this NAD plus is reduced to NADH plus H plus. So this is also involved in similar redox reaction like FADH2. You will see in many reactions NAD is used as a cofactor. In the previously shown examples also somewhere you must have seen NAD and NADP. So so in some cases we have NADP as the cofactor, in some cases we have NAD as the cofactor. The difference, there's a difference between these two. So one is involved in the biosynthetic pathway and one is involved in the metabolism. Okay. So one is involved in synthesis, one is involved in breakdown. Okay. So just a second. Oh wait, you cannot see the screen. Here, can you see? So you see this is niacin, that is nicotinic acid, that is converted into nicotinamide when you have an amide group that is attached to it. Okay, amide group is attached to it. So nicotinamide, this nicotinamide is attached to a ribose sugar and a phosphate group to become a nucleotide, and this nucleotide is attached to another nucleotide that is nothing but AMP. So adenine ribose and a phosphate. This is connected by a phosphodiester bond. So, this is a dinucleotide. So, nicotinamide adenine dinucleotide. So, we have two bases, nicotinamide and adenine, and it is a dinucleotide. Okay. And we have a positive charge here. So, it is written as NAD plus. Okay. We have a positive charge here. So, it is written as NAD plus, and this is the oxidized form, and it is reduced to NADH. So, this carbon will acquire hydrogen, one hydrogen to become NADH, and the plus charge will be neutralized by ring conjugation, and we will have NADH plus H plus. So, there is a gain of proton here. So, that means this direction we are having a reduction, and the opposite direction we will have oxidation. So, NAD plus is reduced to NADH, NADH will be oxidized to NAD plus. Okay. In the second prime carbon, if we have, instead of OH, if we have a phosphate group attached to this oxygen, we will have NADP plus. Okay. So, similarly NAD plus, we will have NADP plus, and the reduced form will be NADPH plus H plus. So, we will have the reaction as NADP plus, this will be reduced to NADPH plus H plus. The difference between NADP and NAD and NADP is basically, one is involved in the metabolism reaction where breakdown is happening, one is involved in the synthetic reactions where formation is happening. Okay. So, NADP and NAD both are cofactors of the vitamin B3, that is niacin, and this NAD and NADP are involved in redox reaction very similar to FAD. Okay. So, this was the last question of the session. If you have any doubt, let me know, otherwise we can end this session now. Okay. So, thank you so much for joining. I will end the session now.